I've seen probably 100 to 200 people send me this exact picture and say, this is proof that the virus exists. Now, here's proof, this one, that the virus is budding off of the tissue. So that's proof that the viral theory is accurate. The viral theory being, you don't see any viruses in the person. And, well, let me rephrase that. There's no viruses, then the virus is attached to the cell membrane of the cells of the person. They do that with the spike protein hooking up with the ACE2 receptor. They inject their business, which is the genetic material, into the cell, reproduce themselves, bud off the cell membrane like this. This is the picture of that. And then you make thousands, millions of copies. Now, I would just caution you that the only part of that you see is this and this. Here is the virus. Here is proof that it's budding off of the cell membrane. That's it. The rest of the steps you cannot see. You can't make a video of this happening. There is no proof except this is produced. Therefore, all the rest of those steps must have happened. So let's take a look at this particle. What do we know about this particle? This is the study that was sent to me. The title of the paper is Appearances Can Be Deceiving. Viral-like inclusions in COVID-19 negative renal biopsies by electron microscopy. This was published in August 2020, so it's pretty recent, in a journal which I never heard of, but is a peer-reviewed conventional journal called Kidney 360. So it's all things to do with kidneys. And my guess is, although I don't know this for sure, that these authors probably were not nephrologists, not virologists, although I don't know that for sure, which is important because that means they don't have so much skin in the game. So now this is going to get a bit boring and tedious, I would admit, but I'm going to read some quotes from this article and then explain what they mean in common English, because this is key to understanding the nonsense that we're talking about. First quote, we have observed morphologically indistinguishable inclusions within podocytes and tubular epithelial cells, both in patients negative for coronavirus disease 2019, as well as in renal biopsies from the pre-COVID-19 era. In other words, those little particles that I just showed you with the black dots, they're saying they see particles that are indistinguishable, identical to those particles within podocytes, I'm not sure what those are, and essentially kidney cells. And they saw them in people who were negative for COVID, and they saw them in kidney tissue from before COVID actually started, which means how did it get there? Because this supposedly was, quote, jumped from bats to humans, or was engineered only in late 2019 or early 2020. So how did these identical particles get there in the pre-COVID era? Next thing, quote, we postulated that endogenous mimickers could be present that are morphologically indistinguishable from SARS-CoV-2 virions ultrastructurally. Endogenous mimickers is exactly what I've been talking about all along. These are identical looking particles that come not from any outside virus, but from you, or the monkey kidney cells, or the fetal calf serum. They are morphologically indistinguishable. In other words, they are identical. SARS-CoV-2 virion, a virion is just one virus, so they are identical. Next quote. Viral-like inclusions consisting both of single vesicles with diameters between 50 and 139 nanometers, as well as packed groups within larger vesicles, were found in all 15 cases, either in podocytes, tubular epithelial, or vascular endothelial cells. In other words, they looked at 15 cases in their kidney laboratory. They did biopsies. They looked at some structure called podocytes or kidney tubular epithelia, or the blood vessels in the kidneys. And in all 15 of these cases, 100%, they saw identical particles as SARS-CoV-2, the same size, 50 to 139 nanometers. But there was no chance that any, quote, SARS-CoV-2 virus was there.